Good evening, everyone. Thank you for joining us tonight. My name is Marina Sharkovska-Tanasovska. I'm part of the Netcetera marketing team. We are honored to have our special guests here tonight, and we're pleasure to welcome our CEO, Karsten Wengel, here as well. So the two presentations are coming up, followed by Q&A sessions. I will invite you to participate in the sessions and make this event more interactive. And now, with no further delay, I will hand over the microphone to Professor Majarov, who will guide us through this event.
Thank you very much. Hello, everyone. I'm Georgi Majarov, professor at the University of Cyril and Methodius. And tonight, I'm going to be your host, moderator of this event. I'm not going to say many words. Now I would like to see a very nice discussion about this topic, which is quite interesting, I believe. So I'm going to invite first Goran Kungulovski, PhD in molecular biology and epigenetics, which will present universal test for cancer detection from cell-free DNA. Goran, please.
Thank you, Georgi. So the mic is fine, right? Yes. Okay. So yeah, thanks everybody for coming in. I will try to be very concise because I'm coming from a bioscientific background, and probably not many of you here are from a bioscientific background, but I will try to make it easy to understand. First of all, I will give you a background of what we are doing, and then I will give you more details about the concept we are developing. So as Georgi said, my name is Goran, so I'm the co-founder of Fingerprint Diagnostics. So it's a company with the intent of detecting disease early and minimize human suffering. The way we want to start is we want to start with cancer, okay? Many of you know that, cancer strikes 50% of men and around 33% of all women during their lifetime, but not many people know that cancer is highly treatable disease, at least when it's detected in a localized manner. So not many of you know, and cancer is quite successfully treatable disease if it's detected early in a localized form. So the success rate, the 5-year success rate is around 90%, but it's getting tricky when cancer has metastasized, when cancer has spread around the body. Then the success rate plummets to around 19-20%. But this, I see it as a positive because this tells us something. This is all right. It tells us that we have an arsenal of tools, we have arsenal of weapons to fight with cancer right now, if we detect it early, if we detect it in a localized manner. And of course, as an extra incentive, not many people know, but for around 70%, cancers causing 70% of cancer deaths have no recommended screening guidelines. And we see them only when the symptoms appear. So this is an extra incentive to be able to devise, to be able to develop tools that would be able to detect cancer early. And of course, the most cost-effective way to treat cancer is if you detect it early. And I assume that if you're able to develop a tool that would be able to detect cancer at least earlier, that would have a phenomenal effect for society and for individuals as well. All of you have heard of cancer, but probably not many of you know what cancer really is. So the way I understand it, so there are they're constantly increasing the hallmarks of cancer, but the idea is that cancer is a phenomenon where cells are basically losing their founding cellular identity. And consequently, they divide in an uncontrolled manner and they invade the body uncontrollably. So this is the way I understand cancer. So since cancer is a disease of identity, of cellular identity, Does any of you know how cells are getting their identity? How cells know what they are? How liver cells know that, okay, I'm a liver cell, I'm not a kidney cell. And it's quite interesting because all of us, every one of us, have the same manual, right, for building humans. We have the same recipe for building humans. We have the same DNA. We have the same info encoded into our DNA. But how could it be that we have tens of types of organs, we have hundreds of types of cells, and we have probably thousands of subtypes of cells? That's very interesting for me. So we have the same recipe, but we have different effects of the recipe. And this is very important for the identity of cells, including cancer. And I mean, as it happens, it makes a lot of sense, because if you take and you linearize your DNA, it will have a length of 2 meters. So 2 meters of DNA in every cell in our body. So this is a problem. So you would assume that the DNA would have to be highly compacted, so around 500,000 times, to be able to stay inside a small nucleus. So the nucleus is around 10 micrometers. So 500,000 times the DNA is compacted. And you could assume that the default state of the DNA, the default state of the manual for making cells and making human beings would be inaccessible, unavailable to read out, because it's very compacted. And there are a lot of processes, so they are called, so by the way, this ragback of DNA, of proteins inside the cell nucleus, it's called chromatin, so this is a very important term. And there are a couple of mechanisms that open and close the DNA, that make available, accessible, or inaccessible the DNA. So as I said, this is the opening and the closing of the availability of the info inside the DNA is orchestrated by chromatin factors. So these are DNA methyl marks, histone marks, non-coding RNA, and so on. And they have the sole idea of opening and closing chromatin to be able to make the DNA accessible. And probably now you can, because many of you here are coming from IT, this is some sort of a algorithm. It's basically a program. So we have the same DNA, but different parts of the DNA are open and closed. Specific parts of the DNA are open and closed in different types of cells. So you have the same DNA, you have the same book, but you haven't highlighted the same paragraphs in the book. So I have highlighted one specific paragraphs in the book. Georgi has highlighted different paragraphs in the book. And this is how basically how cells are getting their identity, including cancer, so all the cells. So that's why I'm saying that this is an identity problem. And of course, since we wanna pinpoint the identity of cancer, we tried thinking about it, and we started thinking about that. Okay, maybe one of these, as I said, one of these chromatin factors that orchestrate the opening and closing of chromatin is the DNA methyl marks. So DNA is built of cytosine, guanines, adenines, and thymines. And it happens that when cytosine is followed by guanine, it could have this, I don't know whether you can see, a small chemical mark. It can have a methyl mark. And this methyl mark leads to different types of effects. Typically, it closes up the chromatin in a very, very stable manner. But what is interesting for us is, so we have around 20 million methylated cytosines in cells. And what is interesting is that different cells have different patterns of these methyl cytosines, of these DNA methyl marks. And of course, we can use them to identify cells. And okay, now we know what to do, but of course, we need a easy manner. We need some easy way to sample the DNA. And the easiest way to sample the DNA is, for example, through blood. And we are in luck here because cells, they divide, they die. As we said, cancer cells, they quickly divide, but also they die, and they release their DNA inside the blood. So they release fragments of their DNA inside the blood. And of course, we thought, okay, maybe we can use these methyl marks in these fragments of the DNA to be able to portray the fingerprint of cancer in the blood. And we know the way healthy blood looks like. We know the fingerprint of the DNA in healthy blood. Now we need to discover the way that cancer's DNA different types of cancer's DNA looks like inside the blood. Okay, and then we started working. So we have an incredible team of domain experts who are basically able to blend the molecular underpinnings of cancer together with the latest, with the most current AI methods and tech. And the idea is to be able to detect the fingerprint of cancer in the blood. Or in other words, so we are developing a platform that would be able to say whether there's cancer developing through a blood analysis, and if yes, it would be able to locate it inside the body. It will tell us where is it coming from. Okay, and then we started working. And what have we done so far? Of course, this is a time of big data. So a year and a half ago maybe, we started working. So we took thousands of DNA methylome data sets. So these were data sets from healthy and cancer tissues, from this public data. So we took thousands of data sets, and then we used them to train an ML algorithm to be able to classify the samples. So this is something like a hobby. We started working on it. It was a lot of fun. And in the end, it turned out that you can see the results were invigorating for us, because you can see, you can even see even if you know nothing about DNA and nothing about science, you can see here in the graphic, you can see a pattern. You can see that different cancers form some sort of different islands. So they form some sort of a archipelago of islands, and they cluster together. So if you can see it with your own eyes, so this would be very easy for a AI or ML classifier to be able to discriminate and discern between different types of cancers. So, and you can see we had great sensitivity, great specificity, so the algorithm worked with very, very high accuracy. And in here, below in the small photographs, you can see different cancers in comparison with their healthy tissue. So they can be well discriminated, and they form different types of clusters. And this was, as I said, quite invigorating. And then we started wondering, okay, can we do the same in blood? Can we, would we be able to detect the fingerprint of cancer in blood? And because this is a different problem. In the blood, you will have now we were working with cancer samples, cancer biopsies, and in the blood, you will have a higher background. And you're wondering whether you would be still able to detect the fingerprint of cancer, because sometimes the prevalence of cancer DNA inside the blood when cancer is developing could be 0.1%, sometimes could be 1%, sometimes could be 10%. It depends on many factors, it depends on many organs. And we started thinking about it. It's not so easy to find publicly available data of cell-free DNA, of blood samples from individuals with cancer, but we somehow scraped some data, and we have some data with individuals with prostate cancer, lung cancer, and colon cancer, with different DNA, with different carried out with different molecular assays, and the results look good. I mean, you can see on the graph, for example, in prostate cancer, in comparison with blood sample from healthy individuals, you can see that they are well discriminated. You have one cluster of prostate cancer, and you have another cluster of healthy blood. And then we tried with another method. We took lung and colon cancer DNA, and you can see that. So these are 2D graphs. But if you see them at 3D, you can even see a better difference. You can see a better line between healthy samples and cancer samples. And of course, this was quite invigorating for us, and it told us that now we have a proof of principle. It told us that now we know how to solve the problem. Now we have the means to solve the problem. And of course, if I want to summarize it, so now we have an ensemble of AI classifiers that are trained on DNA methylome data and that are able with high accuracy to tell us whether there's cancer inside a sample or not. And of course, I mean, now we have a roadmap. We know what to do. We have a proof of concept. We had a successful proof of concept. And of course, what needs to be done is, in order to get a certifiable product, you have to repeat. You have to replicate everything you've done so far in the form of a clinical trial with your own proprietary data. And that costs a lot of money, of course, and this is where we are now. But we have the idea, we have the roadmap, we have a graphic roadmap, but also we have a very detailed roadmap in Excel. So we know what we are doing and we know where we are heading, and now we are discussing with VC funds. We are talking with big partners to be able to take this further. And of course, this is all fine, but you know, there are a lot of people now developing state-of-the-art tools for early cancer screening, and the biggest one is Grail. So they have this is a company from the United States. They have a ready-to-market product now in the States. But I would say this is all right for us because the market is vast, and also they're opening new avenues that cost a lot of money, and new avenues from a pragmatic point of view, but also from a regulatory point of view. So it would be great if someone could open the road for you, could pave the way for you, so you could enter it in an easier manner from a regulatory point of view. But also I think we have the upper hand in certain aspects because, first of all, the ease of use of our molecular assay, and also we are developing an AI classifier that is methylome method independent. Or in other words, we are more scalable because our method could be applied in any lab, so we don't need a centralized lab to be able to carry out the experiment. So we can license them to any lab, and yeah, that could be more scalable to what many of these guys are doing. So they have a centralized lab, and they are accepting sample. And at certain time point, you will hit a limit there. But of course, I will repeat once more: the market is vast, and maybe in the world you can have 100 or 150 companies working on the same idea, working on the same product, and covering different markets, and they would be all well and fine. And of course, here are companies Delphi. So Delphi some of them are using DNA methylome methods, but some of them are using straightforward sequencing of DNA. Basically, they are only sequencing fragments. And we have worked with some of the fragmentomic data, and we can say that it's not so precise and it's not so sensitive as the DNA methylome data, but probably that's why they're focusing only on a lung cancer, and probably it's going to work only for lung cancer, but yeah. And of course, I said before, we have an amazing team of experts, and the team has a lot of experience. So we all have academic experience, but also we have a lot of experience in business endeavors. So we have experience in working and partnering with different hospitals, private hospitals, working with Balkan and European governments, working with NATO military. We were building COVID capacities, and we conducted millions of tests. And of course, after we tackled the COVID pandemic, we want to tackle the next frontier. I mean, it's not the next frontier because cancer has always been the frontier, and this is what we are trying to do now. And of course, this would not be possible without our R&D partners. So we were very closely collaborating with the Netcetera, a couple of people from Netcetera, but also we are collaborating with a couple of guys from the University of Stuttgart, where I did my academic career, and we have some also new data from the university. So something that I had been working on before, and we are quite optimistic that we are on the right track. So now we are, as I said, discussing. We are fundraising. We are discussing with VC funds, and the idea is to conduct a clinical trial to be able to get a certifiable product, and then with the first investment also we want to have a pilot with a forward-thinking government, because this is one of the only ways to put your product to market. So I will close it now. Hopefully we will be able to discuss it further. If anybody is interested in more detail into the scientific part, if anybody is interested more in the business part, the business development part, the market part, I'm here open, and Georgi is here open to discuss it further. Thank you.
Thank you very much. Okay. Thank you very much, Goran, for the presentation. Now we can move to the discussion part, so Q&A session about this presentation, and also after David's presentation, we can go for another round for Q&A session for both speakers and for myself directly, because the technical part of the AI is also in my hands. So please, if you have any question, please go ahead.
I assume you're working with expression data from different kinds of sources. You mentioned RNA sequencing, you mentioned methylation, and I saw something about arrays. Interested how you're representing the data that gets put into the models that you displayed earlier.
So we are using DNA methylome data sets, so we are not currently using RNA sequencing data, because you can use RNA sequencing data only in biopsy samples. So it's very difficult to have meaningful RNA data from the blood coming from the tumor, so it will come only from white blood cells. So in this part it would be, I don't want to say useless, but maybe we would have to develop it further. But the way we are representing the data is, we have the coordinates of the DNA of all chromosomes, right?
Yes.
And the chromosomes are divided by coordinates. So there are different ways to analyze and to feed the classifiers, but the simplest way would be is you have a fragment of DNA with certain coordinates, let's say 1 to 150, and in that particular coordinate you have a certain amount of signal. For example, between 0 and 1. And basically on the left side you have different fragments of DNA with different coordinates, and these can be hundreds of thousands or millions of DNA fragments, and all of them will have a value. Will have a value between 0, and 0 would be the minimum, and 1 would be the maximum. And this is what you do to feed, basically, to train the classifiers. And then you have, I mean, we've been playing a lot, so you wanna be effective with the computer power, with the methods and so on. So we're trying to extract the most meaningful DNA fragments that we know from the bioscientific point of view that have a certain value, and then we use those to train the classifiers. And then even when we train the classifiers, I mean, we can see that some parts of the DNA have a bigger meaning compared to the other parts of the DNA. For example, you can even make a classifier with 100, 1,000?
100, even 100, and you can reach like 85% of sensitivity and specificity, only with 100 methylome sites. So if you compare, for example, with microarray of Illumina with 450K methylome sites, you can even downscale to 100 in order to see very good results in the tissue data.
And why is this the case? This is the case because, as I said, we have around 20 million of methyl sites, right? Around 20 million of methylated cytosines. But the way they work is, the way they have their downstream effects in the cell, they work as clusters. So it's very rare to have one methyl site that will have an effect on the cell, or that will have an effect on the openness and closeness of chromatin. So typically they work in clusters. So there is a cluster of methylated cytosines in the near vicinity, and it has a downstream effect in the cell. And that's why, I mean, we can downgrade it to a couple of thousand sites, and we would lose probably a couple of % of sensitivity and specificity. And this is quite relevant because if you wanna detect thousands or millions of markers at the same time, you would have to do sequencing and so on. So currently, I mean, it's getting a bit more cost effective, but it still takes a lot of time. If you're able to select 100 sites and you're able to devise a PCR test, that could be done in one day. And that would be also ease the way of entering the tech inside all the clinical labs. So now we are currently working with sequencing, but as soon as we have the most relevant DNA fragments, the most relevant DNA sites, we will start working on real-time PCR molecular assays that could be applied basically in any lab. I mean, you can even develop a PCR tech that probably would be able to carry out at home.
Well, if I understood well, on that graph that was with the clusters, each point represents one sample.
One sample, yeah.
And it's actually a vector where each number in the vector is the value, the signal for that gene ID, let's say. Am I right?
Yes and no. So basically, once you have, for example, if you have methylome sites 20 millions, for example, you need to somehow downscale the dimension. And because for some methylome sites you do not have any kind of data, so there are some kind of a missing data, so you need to put on one coordinate, basically to one ground coordinate. So basically what you're doing, you have a lot of dimensions, for example, 100,000 dimensions, so you make some kind of dimensionality reduction using well-known dimensionality reduction technique, and also you're presenting and visualize within 3 dimensions here. But in the normal case, the classifiers basically is making the classification based on those 100,000 dimensions, different dimensions that are already aggregated and incorporated within the different clusters or maybe the different locations of the methylome sites.
Thank you very much.
So yeah, the way I understand it is, I mean, probably I'm 90% correct. So every point is a sample, and in every sample we have taken into account thousands of DNA and methyl markers. So the way it's located in the cluster is based on these hundreds of thousands of markers together, and this is somehow, yeah.
So I'm just thinking from the end game. I mean, let's say you pass seed money and valuation and all this. What do you think could be the use of the test in daily life? I mean, do you think it could be so cheap and so effective that it would be part of a natural blood test you would do when you do a once or twice a year blood screening, which many people, of course, do? Because if that is the effect and the relevance is, I don't know, beyond 90% and so forth, that would be a real breakthrough, no?
Yeah.
Is that the end game, would you say?
Yes, definitely. But I will give you a timeline of what's going to happen.
Yes.
Because currently the market price of similar tests would be between 500 and 1,000 euros, for example. And the only way to implement it would be by working with either with big hospitals or with governments. And this is very interesting for governments because even if it's, in our eyes, it's expensive, 500 or 1,000 euros per test, for the governments it would be economically feasible because if you can detect cancer in earlier phase, so this would be more cost effective for the entire healthcare system. And the way it's going to work is, of course, first you want to apply the test in high-risk people. So this would be probably in terms of cancer 50 years or older, maybe above 60, depends on the government. And then you will get the data. And what is interesting is if you apply, as I said, for 70% of cancer deaths, you don't have recommended screening guidelines. So you have screening guidelines for breast cancer, prostate cancer, in some states for heavy smokers, lung cancer, colon cancer, and cervical cancer. And you have to do the test separately. And we are coming to something that is called positive predictive value. So this is the number of people, this is the number basically of true positives. So if your test is positive, what's the probability of being really positive, truly positive? And probably nobody here will know that if you do, for example, mammography, or you carry any of these screening tests, the positive predictive value is between 10 and 20%, meaning that 80 to 90% of the positive results are false positives, because this is dependent on the sensitivity. It's very dependent on the specificity. That's why you want to have a high specificity, above 98, 99%. So we have a low number of false negatives. And also, it's very important the prevalence of the disease. It was the same case with COVID tests and with the rapid tests. And if you have a low prevalence of the disease, and if you have even if you have the best test with high sensitivity and high specificity, your positive predictive value would be very, very low. And in this case, because you are testing for all the cancers, de facto your prevalence is higher, and this is a main factor in the formula. And now if you apply the test in a high risk group, probably your positive predictive value would be between 70 and 90%. And you are decreasing the false positives. And of course, these require follow-up studies, follow-up diagnostics, and this is very, very costly. But also from the therapeutic point of view. So if you detect cancer earlier, for example, if you detect, I don't remember from the paper the exact numbers, but instead of detecting cancer in the terminal endpoint, if you detect it 1 point earlier, basically you have a betterment of your survival for, if I'm not wrong, somewhere between 15 and 20%. So this is the same as developing new therapies for 3 decades. So this is basically the idea, and that's why governments will love it, because, I mean, okay, they care about people, of course, they want the best for their people, but also they want to be cost effective, they want it to be economically feasible, and this is one way to make cancer screening and later on therapy economically feasible. And that's why I think, I mean, first of all, of course, they will start forward-thinking governments like the UK government. They will start with clinical trials that would be collaborated together with companies carrying the clinical trials. For example, they subsidize part of the test, and they say, okay, we will carry out the clinical trial for 2 years, 3 years, 1 year, and then based on the results, we can decide whether we are going to apply it in routine care or not. And this is the idea. And in the end, probably at Western countries and the States, probably in between 5 and 10 years, this would be part of the routine care. And this will happen, whether it's going to be us, or whether it's going to be somebody else, or whether it's going to be hundreds somebody else's. It's going to happen, and it will become part of routine health care. And of course, it will go, first of all, as a companion diagnostic with a standard cancer screening, and then it will go as a alone tool. what I want to point out, I mean, sorry to be long-winded, but you can apply the same tech. You can apply in diagnostic settings and in post-diagnostic settings. For example, you have you're an MD in a hospital and you have a patient coming up with different symptoms and you suspect it's cancer. And then you want to send them for MRI, for blood work, and so on. But they can also do the test. And they can apply it not for a preventive manner, but in a diagnostic manner. And it can tell them, okay, this is most likely cancer, and please take it further. And also you can use it in a post-diagnostic manner. For example, you want to see whether the treatment is working, because the concept is the same. You are detecting the fingerprint of cancer in the blood, and now you are giving certain therapy, and if the therapy is working, probably your result would be negative. And as soon as you have cancer recurrence, if you apply the test every 3 months, you can know in advance that cancer is reoccurring, and you can give the second round of therapy. And based on some data, based on some statistics data, this would be able to detect cancer recurrence between 3 and 6 months before MRI and the standard diagnostic tools. So this could be quite valuable for citizens. And it would be quite valuable for pharma companies, because if pharma companies do not want to have an early cancer diagnostic tool, they would want to have some companion diagnostic for their therapy. And you know, and pharma companies are quite adaptable. I mean, they will find a way to use your tool, or they want to have, okay, can you now make me a specific classifier for, I don't know, for prostate cancer, or can you make me a specific classifier that would be able to discern between prostate cancer, benign prostate tumors, and other prostate disorders. And if you have the samples, you can do the same. It's the same concept, it's the same training, You can apply the same methods to do pre-diagnostics, diagnostics, and post-diagnostics. So it depends on you as a company where you want to pivot, and depends on your partners as well.
Cool. One of the questions that I had you already answered, but maybe you can go more into detail. So, for example, is there a metric in this field that explains to us or tells us how early we can detect the cancer? Because you said one of the positives about this research that you're doing and this product that you are planning to do will decrease the time before we make an early detection of cancer. So is there a metric that tells us that, how early, for example, the model can detect cancer? And also, which type of data are you using? Because if you're using, for example, cancer data from patients who are somewhere later in the later stages of the disease, does that affect your model, and how can you track that maybe?
So of course this is quite valid, and it depends on the labels you use for training your classifiers. Of course the sensitivity would be different in early cancers and in late cancers. So the sensitivity would be higher in late cancers, of course, and based on other people's data and based on scientific studies. So we expect that the sensitivity would be above 90 or 95% for late cancers, and around between 40 and 55% for early cancers. But also you want to, there is another metric you could use, is that the percent of cancers that would not be detected otherwise. So this is another metric, and this is something scientists use in statistical models, and they really see a added benefit of using similar tests. But from the sensitivity would be from 45 to 95% as you go in the developmental pathway of cancer. And it would be cancer dependent, so certain cancers you would be able to detect with a high sensitivity in an early phase, some cancers. So it depends on the biochemistry of the cancer, on the environment of the tumor, whether it's more amenable to releasing its DNA in the blood, whether it's not. So it would be cancer dependence, but if we are discussing means, so it would be sensitivity between 50 and 95% currently, with the current tech we are having. You know, we are developing tools that would be able to be even more sensitive. And also, as soon as you as soon as you make it multimodal, for example, with DNA methylomes, now you use proteomics, now you use microbiomics, now you use other anthropometric metrics, now you use family history. You can improve the model because this is a simple math. And this is how it's going to get developed. But you have to keep your specificity at a very, very high level. So the bare minimum for me would be 98.5%, because otherwise it does not make any sense. And for the single cancer screening guidelines, so typically they have they're more focused on the sensitivity. So they have specificities between 85 and 95 for the good tests. And that's why, you know, the positive predictive values are so low. And of course, this is an added value if we have a universal test for cancer. But there are a lot of, you know, there are a lot of metrics and parameters you would have to take into account. And as soon as you start working with DNA methylomes, of course, you always would like to update your models if it's economically feasible and if it's cost effective. So you don't want to add proteomics data that it's quite expensive, for example, another 300 euros on top of your sequencing data, and it improves your model for, I don't know, 0.5 or 1%. So of course you want to be economically effective.
We can continue afterwards, after the second presentation. So we have been in the space of epigenetics, and now we are moving to the space of genetics. And I would like to invite David to present to give the second presentation about diagnosis of genetic disorders using next generation sequencing and machine learning. David, the floor is yours, please.
So, hi everyone. Let me start by sharing the slides. It's a long name. But yeah, I'm here today to talk about the way we at Netcetera, together with the people at G-Mendel, which is a Danish startup, have used next generation sequencing and machine learning for the diagnosis of genetic disorders. And I will start by telling you a short story. So in a land far, far away, a child is born, and after a while, it starts showing some symptoms. The parents don't know what it is, so they do the reasonable thing and they go to the doctor, and the doctor recommends a visit to the specialist. In the meantime, they start going on Google, and I don't know about them, but when I go on Google, it's usually always brain cancer and I'm dead in 2 months. But anyways, they go to the specialist. The specialist gives some treatment that doesn't work, so they go to a different specialist, then a different specialist, and so on, until someone finally decides, let's do genetic testing. And in genetic test, this process is still not streamlined, so it takes a while. And finally, after this long journey, the genetic test reveals that the child has some genetic disorder. So the conclusion for this story is that it's a very long journey that takes 6 to 8 years on average, and dozens of medical appointments. So the question that kind of naturally arises is, can we do better than this? Is there a way to improve this whole process? So if we look at this and we see that genetic testing was the thing that kind of is the answer, so why don't we just move it here and we solve the world and genetic problems are gone and the world looks like this. We have flying cars and all is well. So why is no one doing it? Well, no one's doing it because sequencing is very expensive. That's kind of why it's at the end of the line. And it's not affordable to do it to everyone. So yes, but no, sequencing was expensive. It's getting a lot cheaper. We can see here that in 2001, the cost of sequencing the whole human genome was around $100 million, and now it's less than $1,000. So that's like more than 100,000 times decrease in price. I mean, it's 100,000 times decrease in price, yes, but still sequencing of the whole genome of one person is an expensive and slow process. This is kind of how it looks like right now. So there's first a person in the lab that extracts DNA and prepares it for sequencing. Then they send it off to sequencing. And then some specialist looks at the results and makes a diagnosis. And by the way, all of the images are AI generated. So the more you look, the more problems you'll find. Anyways, so we have to improve this process. And we can kind of do a few different things. So first, maybe we don't need to sequence the whole genome. We can maybe only sequence parts that are important to us. That way we sequence less, therefore we spend less money on sequencing. And maybe we can do real-time sequencing and analysis, so we can stop sequencing as soon as we detect a disorder. If a disorder is detected early, we can stop so we don't have to sequence and spend more money. And maybe we can replace this serious specialist with an AI model that doesn't eat, doesn't sleep, and can work 24/7, and we don't have to pay him. Okay, so we started work with this in 2021, and these are some of the milestones that we've achieved so far. So we started in 21 first with chromosomal aberrations, which is a kind of a disease that I'll get to in a few slides. Then we did this, which is a fancy word for sequencing many people at the same time. Then we did this, which is a fancy word for sequencing only special regions that are of interest to us. Then we got IVD certification for a product, which meant that this product can be used as a medical device in real hospitals. And after that, so at the end of 2022, we started with proofs of concepts for many different diseases and ways of detecting diseases. We also did a few other things, which I don't have time to go into right now, so they are left for some future presentation. So, as I said, the solution, the main idea is moving testing from here to the beginning. Well, there's a name for that, and that's called a newborn screening test, basically screening of newborns. But we can also maybe move it even earlier, so before a child is born, and that would be a non-invasive prenatal test. But maybe we can do even earlier, so we can screen both parents and say, if you decide to have a child, your child in the future might have some of these disorders. So we decided to focus on the screening test and said, let's make a product that works on this, and then we'll extend for the other things that I listed here. So Goran kind of went into this, but I wanted to go even simpler because I'm a computer scientist and I don't know biology that much. So here's a simpler version. So we are all made of cells. There's a nucleus in the cells, there's chromosomes in the cells, and chromosomes are made of DNA. And DNA is still kind of complicated, so DNA for me is just a string of characters, A, T, G, C, and so on and so forth. And also worth to mention that people have 23 pairs of chromosomes, one from their mom, and one from their dad. Anyways, moving on. So this is Fivea. It's the platform that we built for the diagnosis of genetic disorders. It has the same three parts where lab, sequencing, and models. The difference is that the sequencing and the models kind of go in the same time, and the models can tell the sequencing to stop as soon as we detect the disorder. And this whole thing is IVD certified as an in vitro medical device and can be used in hospitals. So we focused on two different types of diseases. The first ones are the chromosomal aberrations that I mentioned, and they are types of disorder that's usually because of a person is missing a chromosome or it has an extra chromosome or a part of a chromosome. And the next disease that we focused on are mutations, and mutations, diseases that happen because of mutations. And that's like just one character in this DNA is changed from A to a T, for example. So first, the chromosomal aberrations. We decided to look at Klinefelter syndrome, which affects only males and happens because they have an extra X chromosome, so XXY instead of XY. The next disease was Turner, which affects only females, and it happens because of missing of the one X chromosome. So Fivea, just more in detail now. First, we start with the lab process, which is taking a buccal swab from the mouth, like the rapid COVID tests, then targeting regions of DNA and doing PCR, and then we do barcoding. And what's barcoding? Well, barcoding for me is just getting a piece of DNA and attaching a barcode at the beginning and at the end, basically just like this, to be able to tell us, okay, this piece of DNA comes from this person. And because it's bioinformatics, nothing is that simple. There's also a bit of noise, which will kind of become a problem later, because this means that we have to find where the barcode is in the sequence. So after the barcoding is done, we send the DNA off to sequencing. We use Oxford Nanopore, which is the so-called next generation sequencing. There's pros and cons to that. The pros are that it's cheaper, it can do longer sequences, but the cons are that the errors are a bit higher than previous methods. There's improvement in this in the last few years, but there's still a bit more error-y than other methods. And this is how the sequencing looks like. It's some electrical signals. Anyways, after that, the sequencer starts to give us DNA that we can start analyzing. As the DNA comes, we check the sequence quality and we throw away sequences that are of very low quality. Then we start with the demultiplexing, which is the fancy word for finding where the barcode is in the sequence. And for this, we have an in-house solution that we call Torchlex, which kind of turned out to be about 10 times faster than the state-of-the-art tool Guppy that everyone else uses. And we were quite suspicious of that at first, but it came out, yes, it is 10 times faster, and that's because we use the exact way of how we do barcodes, so we kind of optimized for that, and we managed to achieve some great results. We also detect more barcodes than them, which is the unclassified reads. We have a lot less. Anyways, after that, so we now know this sequence comes from person 11, this sequence comes from person 23, and so on. Now we have to kind of figure out where the DNA comes from. So we see that, okay, DNA number 1 comes from chromosome 1, then chromosome X, and so on. After gathering the data in this kind of way, we have everything that we need to start modeling and kind of feature engineering and some other machine learning techniques of pre-processing the data. After all of that is done, if we just try to visualize, we can see that the data is easily grouped in 2 different islands. So the blue dots are the healthy ones, and the red ones are the people that have Klinefelter syndrome. So this can tell us that, okay, now good, our process works, and we can detect with very high confidence people that are healthy and people that have Klinefelter syndrome. These are the results that we achieved. We have over 98% of accuracy and sensitivity and specificity, and that was basically very good results for us. Anyways, moving on to some different kind of diseases that we support. So we decided to focus on SMA, which is a disease that impacts the ability to move and perform daily activities. It's caused by some mutation or missing of the SMN1 gene, and it's an autosomal recessive disorder, which means that both parents have to be at least carriers for their kid to have the disorder. And here are the likelihoods of each possible way. Anyways, then we decided to focus on cystic fibrosis, which is a disease that has coughing, lung infections, and so on and so forth. And it happens because of a genetic mutation, but this time in the CFTR gene, which is a different gene. And it's the same type of disorder, and the same likelihoods apply as well. So how do we build a screening test for these disorders, and why are they different from the previous disorders that we mentioned? Well, we start by using FIVEA again. Just this time, we kind of plug in a different module for the machine learning part. So the lab, the sequencing, and the preparations, the barcoding, everything is the same. Just the final part is, the model is different. And these, by the way, like the whole product is certified, but then every kind of new plugin that we add for different diseases has to be certified again. And these diseases are, again, caused by mutations, so we have to be able to detect mutations. And this is all of the situations that happen. This green DNA here is a reference of a healthy person's DNA, and then a sequence arrives that has some mutation. We can see here from T to A. or maybe something else can happen, and there's this T is missing here. Or maybe this T is here, but it's shifted to the right, and some A that shouldn't be there appears. Or maybe a sequence arrives that doesn't cover that anyways. So sequences arrive. The challenge here is look at the specific position and see is there a mutation there or not. And this seems simple enough, but the problem is that there's errors in sequencing and the data doesn't really look like this. It would be something more like this. And now good luck figuring out which of the red characters are mutations or errors in sequencing. So we kind of have to do something for this. And there are lots of different ways we tried to detect if a position is a mutation or not. We used some machine learning ways, some ways that didn't use machine learning. And it kind of depends on how many sequences you have. If you have a lot of sequences, you can go without machine learning. If you have very little amount of sequences, you need to have a machine learning approach. And here's the machine learning approach that we kind of settled on using in the end. So we kind of take this data, we transform it to basically be represented as a picture, and then we use some machine learning models for image detection and recognition to kind of see if there's a mutation in the pictures. And we used some convolutional neural networks to say which of the changes in characters are mutations and which ones are just errors in sequencing. For these methods, we have basically our results are more than 90%. Accuracy, and why more than not an exact number? Because these are preliminary results from our proof of concept. We don't have a final product yet, but from our testing we can see that the results are very, very high. So that's pretty much it. We also did many different disorders and many different ways of doing them, but we don't have time to go into them, so I will draw my conclusion here. So the story is, right, we need to move genetic testing from here to here, then the world would have flying cars, and how we do that? By integrating an end-to-end solution that would integrate the lab and the ML, and kind of the lab helps the ML, and the ML helps the lab. And how do we bring the cost down even more? We start by focusing on specific disorders, sequencing the least amount of DNA that we need to, so we don't have to spend money on sequencing, and kind of that's the way of bringing the cost down. This is the team that built Fivea, and basically the top row is the team at Gmendel, the bottom row is the team at Netcetera. So yeah, some of the people here, the green background people are all here, plus Georgi, and yeah, Anna and Lukas are alive and well. They're just in black and white because they couldn't find colorized pictures. So yeah, that's all from me, and thank you for your time.
Thank you, David, for the nice presentation. So yeah, we are continuing with the Q&A about the second presentation first, and we can go just for another round of discussion with the both presentation. Any question?
So how do you align the data sequence to look like an image? How do you do this alignment? Do you just have the whole sequence of all the genes that you have, of like the person, and then you feed it through the network and get your, for example, your solution? Is it like and also which type of problem is it? Is it like a classification problem? Is it regression? What are you getting out of the model? That part I didn't understand, sorry.
We used, it's a library made in Google Research. I think it's called, I can't remember the name right now. Anyways, it takes the alignments and the positions. So they first start by making a pile-up, which is, they take all the forward strands, the backward strands, and they see all of the mutations from the forward strands, the backward strands. They see if, like, they pile up all the A counts, the A forward, like the A base pair counts from the forward reads, the A base pair counts from the backwards reads, and so on and so forth. And they build, basically all of the different pictures are from the different forward, backward strands, and the different, one picture would be all sequences that have the A mutation, another would be a picture that has the C mutation, and so on and so forth. And then they do something that I don't remember, and that's how the model is trained on the pictures, because we have the data sets that it's trained on it's a classification problem. There's data sets of 5 different people, whole genome sequencing, and it's sequenced with, I think, 3 or 4 different sequencing techniques. And the kind of ground truth would be an aggregate of the 3 or 4 different sequencing methods that were used. I'm not sure if that answers your question. I can give you the exact model implementations, and we can go over maybe a bit later. Yeah.
Yeah, I just want to add here. So basically, you have sequences for a specific region, and those sequences have a length from, example, from 500 to 1,200. And first, what you need to do is basically perform the alignment to the reference sequence. And you have a lot of problems here. You have different alignment methods, but you'll face a lot of problem if you have, for example, some kind of structural mutations in the DNA, which means that the DNA part, the sequence that you're trying to align, is not aligning to any part of the reference DNA sequence. And after aligning, you need to have some kind of minimal coverage of each particular region that you're practically interested in. So if you are above 70, around above 70 or 100, let's say, just to round that number, if you're around 100 coverage, which means that you have 100 sequences per specific region that you are interested in, probably you will face with a very simple solution for a point mutation. So we are trying to address whether some particular letter in the sequence is practically located on the specific position. But if you are facing with completely other problems like deletions or insertion or short tandem repeats and structural changes, you'll need to have a really specific model for basically addressing this kind of a problem. And then you have to face with another technique where you are just trying to make some kind of a feature extraction based on the location of the specific letter and also the sequences that is practically obtained from the sample. And we have decided also, if we are not able to do it with this technique, which are quite fast, I would say, in the decision process making, we started with the image, which means that after the alignment on all the sequences that we get from the sample, we are just trying to create the image from zeros, 1 and 0, depend whether it has aligned correctly, that particular letter to the reference sequence or not. And that image that is created basically starts to be part of the sample that will be learned. Practically, one particular region with a specific number of locations represent the sample for the learning process, the image for the learning process itself. And it depends. If you have, here we are facing with the problem that we are not extracting and not making the whole genome sequencing. We are focusing on specific regions that are targeted specific regions based on the diseases that we want to discover, to classify basically. And you need to train a separate model just for those regions of interest specific to this one. Because when you're doing whole genome sequencing or whole exome sequencing, for example, which is quite a lower panel, you're still facing with the problem of low number of coverage. And when you're doing that one with next generation sequencing like Oxford Nanopore, the low coverage that you are facing is very problematic because it has accuracy of around 95 to 96% of a particular position and letter there. And if you don't have a specific coverage there, you're not able to detect that one. But if you are able to build a panel which is effective one and cost effective to sequence that particular panel, basically you are able to learn a specific classifier just for that particular region.
So I can explain it briefly how we do it in the lab. So we have the reference DNA, and the reads that are coming from the sequencer are aligned exactly. And for example, if it's a lower accuracy Oxford Nanopore sequencing, for example, you could see, okay, on the reference I have T and in the sequences I have 70% of the sequences have an A, and the others one have the different base pairs, and you start thinking, okay, this could be true. And then you assess the quality of all the alignments of all the sequences, and then you say, okay, this could be true, and then you want to validate it with a independent sequencing method, because okay, this seems like it's a true call, for example, but we are not 100% certain. Let's apply the old school Sanger sequencing, it's called, and that's it doesn't matter. But you apply it, and if you observe the same result, then you can be 100% certain that what you are seeing in the sequence, in the alignment, it's true. So when you work with clinical samples, this is the, I mean, it's not a rule, but it's a guideline basically. So this is what we are doing. But we are also saying, okay, this is T, and we have A's, and all the A's are red, and we see red. We have a lot of red here, and we start thinking, okay, this could be it. So basically, we are using the same technique with a naked eye in the lab.
Yeah, especially with the point mutation. You're facing with huge problems if you're facing with structural changes, like I said. So it could be some part of the DNA which is completely missing, or just is moved across the different region, or what we have made as a huge challenge, short under repeats, which means that some letters, for example, in some specific genes should be repeated a particular number of times, for example, 40, 50 times, and if it is longer than that sequence of short tandem repeats, it's getting to disease. It is affected as a disease. And of course, it's very simple if you have only those 3 letters that are repeating across the region, but it's not like that. You have another insertion there, you have a lot of interruptions, etc., and that is very difficult to first align and see that there is a sequence that could be aligned with the reference sequence, and then decide later for that one. So it has a lot of challenges here.
We saw 70 sequences, but what are those sequences? Are they from other people, or is that from other people from the same part of the region of the DNA that we're looking for? The question is, what are the other 70 sequences?
You mean the sequences in the picture, the model near the end?
In the same.
Yeah, so usually only one sequence from one person is not enough, because there can be errors. So for every region of DNA, we have like hundreds of sequences for one person. And the challenge here is the cheapest way is to have only one, but you can't have only one. So the balance is to have the least amount of sequences that you need in order to detect a mutation at a certain place. And you need to have multiple sequences, so you kind of have a way to fix the errors in sequencing.
I see.
Yeah, the thing is that basically while you are sequencing, we are able in real time basically to detect the disease, which means that how the sequence is recognized, to which person actually it belongs, to which sample, the barcode, which that sequence belong. We are starting the model, deciding about what we have, and we are certain enough, we are saying to the sequencer, stop sequencing for this particular patient. We have enough sequences obtained for the specific region that we are interested in, so just immediately stop, clean the flow cell, for example, on the Oxford Nanopore device, and just continue with another load for another barcodes and another patients, just to be cost effective. And the price for this kind of a screening technique reduced to approximately 10 to 100 times with the conventional techniques and sequencing techniques that exist currently on the market.
So what I understood is I've worked with Star Aligner, and that takes a reference genome and also a FASTQ read and aligns the FASTQ reads to the reference genome. So is the process that you are doing basically replacing Star Aligner because we want to find where it's not aligning properly? Thank you. And also, once you align with your algorithms to the reference genome, how do you, because you say you stop sequencing after a point, does that not mean that every image would be a different size because it would include a different amount of fragments being aligned to the reference genome? So every image would be a matrix that's a different size. How is that standardized to go into the model?
There are specific aligners for short reads and specific aligners for long reads, and I do not want to go into details. But what we are doing, actually, the sequencing is started. So it generates sequences, and create one particular file, FASTQ file, with how much sequences? 4,000 sequences. And we are taking that file with 4,000 sequences, and we are trying to, real time, to align the sequences, every sequence, create the image, make the decision in the process while the next batch of 4,000 sequences is generated. And if we are certain about the decision for the first 4,000 sequences about one particular region, and we are interested just in that particular region, we are just saying to the sequencer, stop sequencing for that particular patient, basically stop sequencing. Because we have identified and we are quite sure that it has that kind of a disease in that particular region, or it does not have any disease in that particular region that we are interested. So it's basically one batch by batch receiving from what is generated from the Oxford Nanopore sequencing, processing in the meanwhile, making the decision, and just a feedback for the sequencer, stop doing that because we want to reduce the cost, basically.
Thank you.
Yep.
So Goran, you have a lab, right? Maybe that was not obvious for everyone. Would you use the solution that David is making in your lab? The solution of David, I mean the G-Mendel solution, there you would be able to do 100 of patients at the same time, without the person looking into the sequences. So the question is, would you use such a solution, and what would be the cost of one test for one person that would be for you acceptable?
Okay. First of all, for labs, it's important to have the turnover of samples. So if you have enough samples, let's say, I don't know, because this is quite rare. Let's say we have 10 samples a day or 100 samples a day. So this would be quite useful because Oxford Nanopore is faster than anything else. I mean, if you disregard the accuracy, because Illumina is, most of the sequences have Fred score of more than 35, so this is 99.999 something accuracy, and Oxford Nanopore is between 95 and 98, maybe 99 in best case scenario. So, yeah, probably in practice it's a bit lower. Yeah. So if I have enough samples, I would ask, okay, so how many diseases do you cover? And for example, if it's Macedonia, are these diseases relevant to Macedonia? And if they are, I would definitely use it. Why not? But for example, I mean, you said you can use it also in a proactive manner. But if you want to use it in a proactive manner, you would have to cover a lot more diseases. So you would have to cover at least all the rare diseases. I call them the common rare diseases, at least the common rare diseases, because this would be quite useful. And this is exactly the reason why myself and my family and my wife and my baby have done it. But we have done the full sequencing, because you want to know proactively whether you are a carrier of something and whether your partner is a carrier of something. And if together there is a probability of your offspring to have some rare disease, because if you know beforehand, you can do a lot of things to.
So just as an example, if there are 30 diseases approximately.
To safeguard it. I mean, if you're 30 relevant diseases and this is quite cost effective, yeah, I mean, why not? Anybody would use it. So I think, I mean, of course we would have to discuss it further, but I mean, that's the idea now. And this is also, I would think of it would be quite interesting for governments, because now they have a lot of different things for screening different rare diseases. Some of them screen, some of them do not screen. The screening methods are, I would say, a bit of backward. So if you have something like this and you're able to apply it with, for example, every newborn, and you cover, the 30 or 50 most common rare diseases, this would be this is at least my opinion, this would be a significant advancement in the diagnostics. And probably if you cover 50 diseases and this is cost effective, this would make a lot of economic sense for the lab, for the government, for anybody. And although you will have a lot of negatives, rare diseases are probably one of the, together as diseases, are the most common disease probably in the world. Maybe not the most common, but the top 5. I think around 5% of Europeans have some sort of a rare disease. So if you are able to screen for the rare diseases even beforehand or as a newborn screening, this would be an amazing advancement. But probably that would last, screening for 30 or 50 diseases as a tech would last a couple of years. And then you have to devise something that would cover more diseases. And in the end, you would want to have a cost-effective or economic test that covers all the diseases. For example, typically, let's say at least whole exome sequencing. And whole exome sequencing now you can do for a price. If you are a lab, for between 200 and 300 euros or dollars. But this would be, as I understood, this would be something like 20 euros per sample. And I really like the Oxford Nanopore sequencer because in comparison to all other sequencers, you can do easily library preps. You can do the sequencing in one day, in a couple of hours at least. And with the other sequencers, if it's not highly robotized, if it's not highly automated, so it takes a lot of time. It's complicated. You need highly skilled personnel to carry out the work. It's tricky, and of course, you have to cover the entire sequencing cycle, and then you're getting the results, and then you can start analyzing the results. And in here, you analyze the results in real time, and that's why probably you have the cost effectiveness. But of course, the flow cell you can reuse only a couple of times. So then you are losing the number of pores. But I didn't know that you're using basically a localized browser snaps basically, and you train your model on that. I was always wondering what exactly you're doing. And
so now you know.
and it's quite interesting and intuitive as well.
Okay, last question.
And what I wanted to say, we've worked in diagnostics in my lab, and these people, their stories are heartbreaking, really. And you are right, they have a diagnostic odyssey of 6, 8, 10, 15 years, and they are getting out with nothing, and they would have to take the entire clinical pathway, and they are still getting out with nothing. And there's also some statistical data stating that if you do the DNA test as a first step, this would be more economical for the society as a whole. And of course, for these people, you're basically cutting their diagnostic odyssey. And for many of the rare diseases, you don't have any treatment. For some of them, it's time dependent. For example, for some of them, if you detect it in the first couple of weeks of life, then maybe you can have a normal offspring. But if it's a bit too late, you have irreversible problems. And this is, I cannot stress enough how important this is. Although, there are therapies for only 5 to 10% of these people, most of them will take their comfort of knowing what's going on. Because we've had people who had been undiagnosed for 30, 35 years. We had one sample, they didn't have a diagnosis for 35 years. And as soon as we did the exome sequencing, we found one variant. And the variant was reported in a paper 2 months prior. So if it hadn't been reported in the paper, we wouldn't know. Yeah, so it's this is how you drive science. This is how you nibble on the scientific pie. And if you have if you have a lot of data, and this is a zeitgeist. This is a time of big data. This is exactly what you need, and many other people and scientists could use your data to train different models to do quicker diagnostics and so on.
So can this type of technology detect so-called pre-cancer cells, such as cervical intraepithelial neoplasia, grade 1, 2, and 3? And whether can this algorithm predict whether the cells will transform definitely into cancer?
If you have the right samples to compare with, to create your classifier, no problem. So now there are even tests using between 5 and 10 markers that could make a really accurate assessment whether you would go from scene 1 to scene 2 to scene 3, and whether it would progress further. So this is definitely going to happen, but I would like it to happen as a part of one whole, of one test, so you would not have to spend the taxpayers' money for many different types of tests that basically do the same thing in a very focused manner.
And will this test be superior to regular Pap smears?
Yeah, because Pap smears are very inaccurate. So they have a low positive predictive value, they have a very, very low sensitivity, and they're basically on the border of a, I mean, I don't want to say a guess, but they're slightly above guessing. So, but this is the standard of care we are having now. So, but yeah, I gave you an example about prostate cancer. The same example could be given for cervical cancer. If you have the right samples to train your algorithm, yeah, definitely.
Thank you.
Thank you very much. I'm going to close now the official part of the event, and I would like to invite for some drinks, and of course we are going to have some networking session. We can discuss, and we can communicate in a more relaxed atmosphere. Thank you very much.